Hello, everyone. Thank you for inviting me to speak at this year's Congress. Over the next 20 minutes, I'll be discussing the Culturomics approach, how we started it, and its significance in the field of clinical microbiology and infectious diseases. So let me start by summarizing the common methods for bacterial identification in routine labs. We'll start first by the old commercial and molecular approaches. One of the most typical and oldest methods implemented in bacterial identification is the phenotypic and biochemical approach, which relies on multiple factors such as the enzymatic activities, acid production and utilization, carbon source, fatty acids profiling, as well as micronutrient utilization and metabolism. Nowadays, there are commercially available kits that can easily test for each of these factors. These kits may not be the best option when it comes to identifying new and closely related organisms because they lack specificity, are time consuming, and not cost efficient. On the other hand, these biochemical and phenotypical characterizing kits can be informative once new bacterial species have been isolated and identified via other techniques. In addition, most clinical microbiology labs still use chromogenic media for pathogens targeting in clinical samples due to the availability of a wide variety of inexpensive selective and differential media. However, the low sensitivity, the uncertain time of bacterial growth, and inconsistent culture handling remain a challenge. As a result, this approach requires the use of complementary molecular techniques to confirm the results, specifically when media are used to isolate bacterial complexes and identify them from human specimen. With the advancements of technologies, molecular tools were developed as a faster, more specific, and more sensitive approach for bacterial identification. Probe-based methods such as DNA microarray technology that targets different organisms showed to be efficient and cost-effective in pathogenic and known organisms identification. Nevertheless, amplifying and sequencing methods like 16S or RNA gene sequencing revolutionized the field of clinical microbiology in the 20th century with their ability to identify what has been previously called uncultivable bacteria. This sequencing approach started a new era in the field, specifically in human microbiome description and clinical sample analysis, where it rendered the identification of certain pathogenic organisms and other unidentifiable species possible. Although 16S RNA gene sequencing provided great advantages, it failed to identify certain bacterial species. Therefore, this approach was complemented by 23S RNA gene, RPOB, and real-time PCR for intracellular bacteria for an optimal identification of bacteria at the genus and the species level. Now, specific gene sequencing was further developed into a much more efficient approach known as the next generation sequencing or NGS, which enables the identification of bacterial species by generating draft genomes from assembled contigs or even nowadays by analyzing complete and closed genomes. Most commonly used technologies in this field are the Sanger sequencer, Illumina sequencers, PacBio, and nanopores. Despite the advancements of sequencing approaches and the presence of a large sequence database, an NIH nucleotide, these methods remain time-consuming and expensive relative to other techniques such as the MALDI-TOF MS. MALDI has recently revolutionized our capabilities of bacterial identification in terms of efficiency, specificity, and cost. In addition, this method proved its importance as a first means of identification in clinical labs, particularly with the establishment of a new database which contains a significant number of spectra that correspond to the commonly encountered bacterial species. This approach supported the Culturomics pipeline as well, which relies on massive and rapid identification of large number of isolated bacterial colonies. In fact, while analyzing the microbial content of the human gut, MALDI-TOF assisted in the fast identification of bacterial species and in filtering potential new organisms. This technique modernized the concept of bacterial identification in clinical microbiology labs by successfully identifying a significant number of pathogenic and fastidious bacterial species in minutes, including anaerobic, aerobic, gram-positive, and gram-negative species. Furthermore, it helps in identifying toxin, antibiotic resistance profile, archaea, eukaryotes, and viruses. The ability to quickly identify pathogenic or potentially pathogenic species in clinical specimens, specificity, and low cost justify why this technique should be more commonly applied in the scientific community. In doing so, this will expand the existing database and in turn decrease the limitation of other techniques. Now, with the advancements of the bioinformatics techniques, the metagenomics approach emerged, which is a culture-independent approach that directly targets the DNA present in any environment. This method has resulted in a huge amount of data, especially when used in describing the microbiome, human, animal, soil, and so on. Metagenomics played a major role in revealing the bacterial compositions of several environments and their impact on health and diseases. Moreover, it has been implemented in different aspects in the field of infectious disease and diagnosis. Yet, it soon appeared that these molecular techniques and bioinformatic tools have multiple drawbacks such as depth bias and inability to detect, in some cases, the causative pathogenic bacteria, such as the investigation done into the Shiga toxigenic Escherichia coli 0104H4 outbreak, or the Campylobacter and Salmonella and diarrhea cases. All these has been reported in several studies. In addition, incomplete genomic databases make it difficult to assign a precise taxonomic rank to a substantial number of sequences. Most importantly, these methods do not provide a pure culture of microorganisms, which is crucial for further strain characterization in vitro models or host interaction studies. Also, when utilizing these molecular techniques, scientists were able to see a large amount of unreadable sequences, in other terms, unclassified sequences that correspond to organisms that were never been cultured. And these generated data made the scientific community believe that culture would no longer be needed. This is why we wanted to define a new approach called Culturomics, which was introduced with the vision of optimizing culture conditions in order to show that the term uncultivable organism is misleading, since all microorganisms can be cultured when using the right conditions, tools, and routes. Besides the different culture conditions used, this approach is coupled with an efficient, fast, and cost-effective identification methodology based on MALDI-TOF MS and 16S rRNA gene sequencing in the event of MALDI-TOF MS identification failure. Now, the main question is, why do we need to culture these organisms? It's because the molecular approaches do not tell us if the organisms detected are living or dead bacteria. At the same time, we don't know how representative it is. It might be a contamination, it might be a passenger bug. Also, the high amount of OTUs prevent us from developing bacteriotherapeutic approaches or to be able to analyze the clinical specimens in a proper way. And the reason why we need the living organisms is simple. I'll give you a couple of examples. Akkermansia municiphala, which was isolated in 2014, showed to be correlated to obesity. It has been decreased in stool samples collected from obese patients, which means that the replenishment, and this has been proven also in mice models and currently it's present as a probiotic, that the replenishment of this bacteria can support the obesity symptoms. Same for the H. pylori in gastric adenocarcinoma, Faecalibacterium prosnitzi in IBD, Clostridium butyricum in necrotizing enterocolitis, Enterococcus in PD-1 treatment, and others. All these say that we need the living organisms in order to study it, in order to culture it, to be able to have reference for it, and see what's its biological role, if it's pathogenic, how to treat it, and so on. Now, before I move to talk about the role of culturomics in clinical microbiology, I want to talk a bit about the pipeline of culturomics, which was developed during our effort to describe the human gut microbiota compositions. Basically, samples are collected using special transport media to protect anaerobic organisms from oxygen. Part of the samples collected usually goes for sequencing by metagenomics and other goes for culturomics. Using anaerobic chambers, samples are incubated in defined medium and then follow-up are performed over 10 days. Colonies are picked and identified using MALDI-TOF-MS. Unidentified species are subject to 16S, and novel organism sequences are compared to operational taxonomic units generated by metagenomics. Correlation is made, and novel organisms are deposited in international collection banks. Now, the main question is, how did we choose the defined medium, or how we optimized culture conditions to be able to isolate organisms based on culturomics to be efficient and support the pyrosequencing approaches, or to overcome its drawbacks? Mainly, we tested more than 240 culture conditions that imitates the gut microbiota environment, which can also be done in other samples, in type source of samples, and we have done it as well from in the environment, skin, oral samples, and so on. So we usually look at the challenges, like the growth of bacteria having different physiochemical properties. So what we try to do is test various temperature, different atmosphere, aerobic and anaerobic, including anaerobe cabinets and microaerophilic cabinets. Also, we try to inhibit the overgrowth of certain organisms to be able to allow the low abundant organisms to grow. This is usually done by targeted antibiotic use in the culture media, filtration, phages, phage elimination, or phage addition in certain cases, as well as thermic shock. Also, we try to pre-incubate fastigious organisms in a mix of a rich culture media that contains defined biochemical set of molecules that allow the growth of targeted bacteria from a specific phylum. All data regarding the culture conditions and its optimization has been published online and can be checked for adaptation in clinical microbiology whenever we are looking for fastigious organisms and we have a confirmed case of infection but culture negative results. Now, as a result of using culturomics in the Human Gut Microbiota Description Project, we were able to isolate more than 1,700 species from stool samples, of which 450 were novel organisms that scientists were never able to isolate, and most of the time sequencers were not able to detect it because of its low abundance, or if detected, it was classified as unknown organisms or an operational taxonomic unit that correspond to an uncultured bacteria. This effort supported the expansion of the gut microbiota repertoire, and among these 450 novel organisms, 102 organisms were found but not identified in previous metagenomic studies from the Human Microbiome Project. We were able to attribute around 20,000 orphan genes, and a significant number of the isolated new bacterial species were detected in metagenomic data as OTUs in studies aiming to describe the healthy gut microbiota. Now, the novel organisms that we isolate, we tend to perform something called taxonogenomic description, so we can report its isolation and discovery and share the data with the scientific community, especially among the clinical laboratories. So in case it was encountered in a clinical sample, they can know what this organism is and what's its antimicrobial resistance profile, what virus genes it expresses, and so on. To do so, we perform genome sequencing, we do annotations, we target specific genes, virulence genes, AMR genes. We perform phenotypic characteristics and biochemical analysis in case scientists want to use APIs, for example, to identify these organisms. Or also we do proteomic analysis in order to create a mass spectra that can be added to the MALDI-TOF-MS databases. So if scientists are using this machine in their clinical labs and it was encountered as part of a clinical sample, it can be identified in routine basis. Now, the first role of culturomics in clinical microbiology was the optimization or the enlargement of the MALDI-TOF-MS database, which is becoming the gold standard in clinical microbiology for bacterial culture identification. And this has been done by the addition of thousands of reference spectra to the databases provided by the manufacturer. The ability to have a very large database support clinical laboratories to identify organisms at a fast and low cost, which is around 1 minute and 46 seconds per colony at a cost averaging between 1.35 to 1.43 euros in a very accurate way. The database has allowed in 2018 to identify 64 potential pathogens with 14 different new species that were isolated previously by Culturomics. On the other hand, and as mentioned before, Culturomics supports the isolation of novel organisms and the ones that are low abundant in the human microbiota. Thanks to the isolation of these novel organisms and the description, the taxonogenomic description of these species, which includes the addition of the spectra of each organism into the MALDI-TOF and the submission of the genome 16S and all information about these species online, 21 novel organisms were identified 105 times in 98 patients in a retrospective study done in France. This means that clinical microbiology labs are likely to identify what is already known to exist rather than discover unknown microorganisms. So Culturomics supported this by providing new culture techniques that allow the isolation of fastigious bacteria and anaerobes from polymicrobes. Now, this is a list of examples of novel organisms that we isolated by applying Culturomics on clinical specimen. For example, a new species of actinomyces was isolated from breast abscess and urogenital infection. New species of Bacteroides, Pseudomonas, Corynebacterium, Nosocomicoccus were isolated from blood cultures or bacteremia episodes. Now allow me to end my presentation by saying that Culturomics and metagenomics demonstrated the significant role that the bacteria play in both health and diseases, including conditions like colorectal cancer, IBD, and obesity. These findings open the door for closer collaboration between infectious disease specialists and other medical disciplines, encouraging a shift in perspective to view bacteria not as source of infection, but as potential therapeutic agent. The shift is further supported by the success of fecal microbiota transplantation in treating C. diff. However, the limitation of FMT drive us to refine Culturomics and develop defined microbial communities with targeted therapeutic effects, potentially offering a more precise alternative. Thank you all for your attention, and I'm happy to receive any questions.